Hey, thank you everybody for joining us. Today is February 27th, 2026, and thank you for joining us for our second episode of the Cannabis, Hemp, and Natural Psychedelics Committee webinar series. My name is Tui Vu, and I'll be hosting today. Before we get started, I did want to read the after webinar disclaimer. The views and opinions expressed during the virtual event are those of the presenters and not necessarily the Association of Food and Drug Officials. Participation in the virtual event does not necessarily constitute an endorsement of the speaker's views, products, or services. This virtual event cannot be used to train AI models. And so we will go ahead and get started. I wanted to welcome Dr. John McKay. Dr. McKay is a recognized educator and scientist and the founder and CEO of Synergistic Technologies Research LLC. Dr. McKay has held leadership roles in Newbridge Global Ventures and the Waters Corporation. He contributes to scientific literature as a journalist and editor for Extraction Magazine and Terpenes and Testing Magazine, and also serves as an assistant professor for the Robert Lerner MD College of Medicine, University of Vermont. He holds a PhD in chemistry from the University of Vermont and a bachelor's in chemistry from St. Lawrence University. Dr. McKay has been recognized with numerous awards, including the President's Club Award and the Cannabis Scientist Power List. He holds several patents in extraction of cannabis. Dr. McKay and I go a long ways back, and we were saying at least a decade because we used to run the circuits together for the expo. So very excited to have you with us, Dr. McKay. And I will hand the screen over to you.
Oh, thank you. And I will try and share my presentation. Does that show up? Good.
Yes.
Thank you very much. And it's an opportunity to share some things about extraction, but also being able to reconnect with Twee on some of the stuff since I think early 2010 when she had different roles in Colorado for regulatory and looking at all the different parts of the safety of this industry early on before it was even um, a large part of the world that we see today. And so on that side, I'm really talking about the principles and practices in assessing compliance of cannabis manufacturing. And one of the things that I see is as any trilogy that you have, whether it's The Hobbit or whether Lord of the Rings or whatever trilogy, you're always starting off with the first part of getting used to the characters, where's the story going to go? And this part of that trilogy gets us to the basis of extraction and separation, which is more towards the concentration. And so looking at the different parts of that, most of the time people are a little more anxious about, I'm a little more anxious that my slide didn't move forward. So if I move by the end of this session, looking at some of the different modes of extraction, separation, hyphenated processes, and being able to take those parts and being able from there to make evaluations of different parts of the cannabis industry. Not much different from any other natural product, but still we want to be able to look at these processes and evaluate them on the base level. And from there on the next two sessions, going into a little deeper of some of those facets that we want to see. And from there, being able to apply the knowledge of separations. How do we have to evaluate a facility? What's new in this technology versus what other technologies are showing? And then applying that to the conversations that would be happening in these regulatory manufacturing sites, and then applying the gaps that go in between. I use the analogies most of the time of what happens within our own traffic world. You come into a town and suddenly the speed limit has gone from 75 to 45, or it's gone down a street that you think is very straight, and why aren't I going 50? Each one of those parts has been able to have a speed limit that was determined by someone based on there being housing. Maybe you don't want to go down the street at 35 miles per hour, but that's the speed limit. And within that, you don't want to be going 75. And if you're through a school zone and you may not agree with it being 25, it's still the regulatory. And so within this industry, sometimes we find that the manufacturing is going, I don't know why this restriction is being here. Why do I do clean in place, or why do I have to know more knowledge of something within a manufacturing facility? It's because they know that it's a school zone. They know that you're being able to be guided through what the regulations would allow you to do and the safety of this technology. So this is not the first natural product that's ever been out there, all the way from the Pacific yew tree to fungus with cyclosporin. Each one of these has gone through a process to be able to evaluate the components that are in that natural product and then finding the activity sites, and from there being able to use those as a medicine or as another ingredient into a food product. I mean, anytime I'm having pain, I'm not typically gnawing on a willow bark. I would typically take an aspirin that I might find at a local store. And those development sites are through well-known and through regulatory world of natural products. And so the very first thing that you would see in any natural product where you're finding that it's made a difference in some sort of facility or some sort of country, that you're seeing it's being used for something that would be able to alleviate pain, or you see a site that allows you to have a positive experience. You typically are identifying the unknowns. You do the elucidation of different structures. You isolate, you purify, you find out the mechanism of action that happens within each one of those. And then from there, you move towards product development. How do you go and use this product? What are the ingredients that would be allowed to be within that? The method development, validation to make sure that everything that you've done is actually equal to what you believe it should be. And then you have quality control. You have identity. You have purity. You have strength. You have composition. You have process monitoring. All those things are very critical when you're providing a product that is being used in the human body or a mammalian body. And after that, you have post-market, and you look at adulteration or things that can happen, or contamination, or label studies to be able to know how long it's a shelf life, how long will this product be in the form that you brought it into, and what would be the types of things that you would ask that user who have you purchased to for storage. Should it be in a dark place? Should it be cool? Is it allowed to be out in room temperature? So many products that you see, typically you have that. Milk is a good instance, for example. You're not going to leave the milk out on the shelf. You're going to have it in your refrigerator, so there is some refrigeration so that you're able to not be able to make bad products through that, even though you started off with something good. And those techniques and those tools have been well thought out, and that's through productivity, compliance, and throughput. Each one of those is comparative in the commercialization of natural products. And I'm often asked what's the best concentration of these any products, but certainly in the cannabis industry. And within that, I have this world of seeing best. And for me, it's the botanical materials, no matter what they are, that you maintain the integrity of that material and concentrating in a forms that you want it to be in. And we'll discuss, how do I regulate that? How do I know which ones they are? And the next one is effective concentration with efficacy. Efficacy is first. Is it safe? Is it what it's supposed to be doing? Efficiency and economics come along when you're being able to make sure that you're having something that's profitable, that allows you to continue the research, to allows you to continue to bring that product to who you want it to be going through predefined boundaries. You define the boundaries and then you make them. And then we hit on what we're talking about today, along with the top two, and that is safety and the safety of everyone throughout the processes and throughout the manufacturing. So it's not just the manufacturing, it's not just the product. It's also looking at that opportunity of, pesticides or any other type of um, uh, things that would be on the plant or mold or anything that would be based on the plant that the product has been sitting out. And so you want to have the safety and being able to assure where those are. And then finally, testing with modern technology in every facet of the process. And so we'll talk about those also over the next trilogy. But I find most of the time people are doing extraction and they're not really knowing that they're doing extraction or concentration and based on two different principles. So I like to start off with something of coffee. Most people are making coffee in one form or another, or buying coffee or tea. And each one of those is a product that you're being able to have that you want in your extraction. And most people are saying, well, why would I have tea? Most of the time, you're also saying, I want the caffeine. I want to wake up. Well, coffee has a lot more constituents than just the caffeine. If you just wanted caffeine, you would take something with just caffeine in it, a caffeine pill. But most of the time, we're looking at how do I get this coffee out and enjoy it? So one of the things that we do for coffee is it depends on the different solvent that you have for making coffee. And this is an example of something that you go from cold brew coffee versus regular iced coffee. And when regular iced coffee is made, you first make it through high temperature water, making coffee like you regularly would. And the other one is being able to start off with cold water. So both of them start off with water, and they both start off with a process of a coffee ground that's a little bit different from each side. But I'm being able to go back through, and we see that hot water between 195 and 205 is bringing out different compounds in place. Next slide. And as you go through this steeping process, you have the ability of making the coffee in a few minutes versus a long steep where you're having coffee over at least 12 hours. And that's because you have a different phase of water. They're both water, but you have it in a different phase. One is with heat and the other one is with room temperature or cold. And within that process, you're having different compounds that come out. And so when I look at the next slide and I see that when I'm getting to that next step, I'm filtering. And so I filter out the coffee grounds from each one. And when I'm doing both of those, are seeing the same coffee grounds. And when I look at the next slide to see what I have for a product, I let it cool and then I add ice to it. Where the other one, I have it's already cooled. And when you do those two different separations and extraction, you're having water that you now have 1,500 compounds in a hot coffee and about 1,000 compounds in a cold brew coffee. And just because you've cooled it down so that they're at the same temperature, when you come back to that coffee in the refrigerator a day or 2 days later, and you have the iced coffee versus the cold brew coffee will still be tasting great for another month. The hot coffee will not, because it's taken in another 500 compounds that slowly degrade it. So even within those two processes, we're doing concentration, and extraction is a liquid or a liquid solid separation extraction. And so when you have the filtering is a separation, and we'll look at those in more detail in a moment. And on the next slide, when I'm seeing that process, it says so what? We're providing material for making coffee, but you're not really making coffee. You are extracting coffee grounds into the water. And you also have different parameters that were made on both of those processes to get you down to that final product that you're using. And it depends on whether you're a home or a commercial facility. If you're at home and you're making your own coffee, you can change out the different grinds. You can change out the different types of coffee bean. You can change out how you want to be making it on a fine grind or a thick grind. And when you're in a commercial facility, they're already dictating on what you would have. So you're going to have a product that is equal every single time. And you have steps in chemistry behind each side. So how does this differ from an instant coffee? When you have an instant coffee, you already have something that's been made. It's already been into a coffee, and now you're just adding water. With the other ones, you have with the instant coffee, you don't have any more parameters in how much coffee you add into that cup and then add the water. You have those two parameters. When you have the parameter where you're actually making a product, now you have all the different facets that you can change. And on the next slide, when we see how you can change those, what's different between an espresso versus a regular coffee? Pressure, because the particles are so small that that's what you're doing for an extraction. You need added pressure because it turns out gravity is not strong enough to force that water through that mixture that you've already ground. And the ingredients, did you put things in before? When you have the coffee that you might get somewhere and it says vanilla or something, you might already have sprayed vanilla onto it, or you may have already put other things into it, or you add them afterwards. So an added ingredient, you're not typically going to extract coffee with milk, even though you might add milk after it. So it's the sequence of events that you put into that extraction, into that concentration of the coffee. What's the shelf life? The shelf life depends on how you actually made it. And each one of those, you have steps in chemistry. When I look at the next slide and I see the nomenclature that we want to be able to use when we're talking about concentration. Extraction is done with a solvent. It's done with a liquid involved. Separation is without a solvent. So there is no real definition of solventless extraction. It's a separation. And to use those terms correctly is also important, but that doesn't mean that the rest of the world is following your nomenclature. And when we look at the next slide and I see what does it matter? It's so what is your goal? What is the goal of what you're trying to do and what you're trying to concentrate? And do you do it the same way every time? And so when you do it the same way every time, then you do have a standard operating procedure by definition. Even if you get up in the morning and you put the same amount of coffee into your filter, you put the same amount of water through the filter, that's a standard operating procedure. But if you're varying that and you put in a different grind, a different coffee, a different flavor, then you're going to have a different result. So is it historical or is it a mem? And we'll look at that next. What is a mem on the next slide? So if I look at, you go to the next slide. And so we're going to look at these processes over the trilogy. So I have a process parameters. I have facilities and equipment. I have materials and flow, and I have documentation and records. All of those come into play when people are looking at how do I maximize this opportunity? How do I look at what this process is? If I look at the next slide and we focus on just the first one, we'll look at process parameters. I will use this as a template for what we will get from the other parts of the business, because when we're doing that, we're seeing that these are the questions that should be asked. What's the depth of the knowledge and experience of the person that you are talking with about the manufacturing? What exactly is being controlled? And how do you know? That's a great question for someone. Why are these limits selected instead of others? What evidence shows the process remains in control of what you had from before? And what happens when it's not in control? With those few questions that you're talking with someone and you're evaluating a manufacturing facility, each one of these will come to control. Each one of these will allow you to understand the depth of the knowledge and the being able to know that they have a process in control. If you don't know what's being controlled, whether it's temperature or the type of solvent, the amount of material, or the material or the factors that are in the material, each one of those is going to have a reason for making sure that they know what they are supposed to be knowing. What are the limits? Each one of these will go back through, and then the evidence that the process is in control. I know that I'm getting the same amount of material. I know why I'm getting the same amount of material. And if I look at the next slide and I find out what my inspection intent is, the questions are allowed there to be able to understood if the process is understood, if it has boundaries around it, or whether it's just being merely repeated on something that they had heard or something that they saw in a rule book, and not really understanding that if something does change, that they know why it changed, so that you have an out of spec responsibility. And the red line on this on the next slide is that they don't know, and there's no cause and effect. They cannot understand that something's out of control. What do I have to do to fix it, or what should I do if I see those things coming out of balance? In the next slide, we're going through and we're going to start to talk about chemistry. So if you thought chemistry and theory were hard before, this isn't going to change your reality of the universe. The chemistry is still there. Physics is still there. It's still going to be the same, but the goal is to be able to look at that view and change your opinion about what's being done and being understood by yourself, or also the people that you're interviewing. And on the next slide, when I look at that, there's three things that come to play: phrases and phases. We use a lot of things, typically, like even the first slide that I spoke about, of I'm going to make coffee. Well, you're not making coffee. You're actually making a liquid that has a concentration of the components that you want out of that coffee. Fiction and facts, things that people have heard, things that people have believed through the years, and what's really the case and how those happen. And the second one is colloquial conversation versus correct chemistry, which goes back to the very first one of there is no solventless extraction. That is a separation. And so being able to use the terms correctly and understanding the influence of each one of those formulations is important to know as we're moving through it and looking at evaluation of different processes. And compliance. In the next slide, when I look at each one of those things, it's narrowing that gap. What is that gap, so that we're able to have a communication and we're able to talk about the differences between nomenclature, the differences between how do I know what a liquid extraction is versus a liquid solid. Each one of these is important because it has a tantamount formulation impact, and the amount of dosing, and the active ingredients and the inactive ingredients, and having things that you're not wanting to have into a formulation. On the next slide, when we're talking about MEMS. MEMS are things that happen. Go back through and you still look at a MEM. And a MEM is an idea, behavior, a style, something that begins with one person, then continues to grow and continues to have more and more impact. So for me, it's a few good memes, and like the movie with Jack Nicholson, you can't stand the truth. And if you look at that, you have to be able to move from that so that you're able to have a conversation about chemistry versus the conversation about what people believe or have used in a different way. In the next slide, we talk about how are these processes followed? Why is this process filed? And what is happening with compliance? Compliance is incredibly important. And compliance happens the same way that we talked about with the speed limits. You're right, you have a straight road. You're right. It looks like there's nothing there, except it has a 25-mile-an-hour limit because there's a school. You have processes in place that need to have the timing and to be able to be complied with, even though it doesn't make sense on why they would do that. It's not really about that, because you have rules and regulations, certainly through the regulatory on botanicals and other natural products. Those things are processed to be able to make sure that there's not bacteria, that there's not mold, there's not other things that have been from the previous extraction. You're having all the clean in place. You have so many things that allow us to be able to go through and make sure that that product is compliant. There's many people that are doing this extremely well. but there's other processes that we will come into play that haven't met those criteria and still need to be able to be evaluated and talked about. This is why we have this in place. In the next slide, we start to look at the theory. And I don't want to go through this theory, but there is very strong chemistry behind why like dissolves like. And what that means is if I have something that's water soluble, it's a hydrophilic, which means it loves water. There's a reason that those compounds go into that one. The typical one that's always used is oil and vinegar or oil and water. And so you know that sometimes something will go into oil, but it won't go into water. And so you have all the chemistry that can be done. It has a scientific fact behind it, but we're going to go through a different phase. If I look at the next slide, I would prefer that we talked about things that are more done within a sequence. And so we'll talk about in this presentation, as well as the next one, the preparation of a feedstock. And I go back to coffee over and over again. And that is, we don't take coffee beans and just throw them into water. We grind them, or we have a way of wetting them. We have a way of being able to know that they are ready for the extraction process. And then we have extraction conditions themselves. And there's a lot of things that come into the extraction conditions as you go from a greater and greater compliance and a greater and greater looking at different compounds that you are taking out. It can be as simple as pressure, temperature, solvent to feed ratio. What does that mean? It means how much water and how many coffee beans do I have? How much water am I going to use to the ratio of coffee beans? And then also you have a superficial velocity. What is the actual velocity of the solvent going through the coffee? We know that it doesn't go exactly through the coffee, and we also know that there's different conditions. And then afterwards, you have separation. So the separation could be the filter paper. It could be the temperature that you have. If you want to do a simple experiment, you can do the following. And that is when you have a coffee maker and you pour water through it, no matter which phase it's in, and you were to take 12 cups of water and you have a certain amount of coffee beans that you would use, you would have that hot water go through, grab that first 8 ounces of water and of extraction, and take that coffee cup and put it off to the side, and then continue through the rest of the extraction. If at the end of that time you've gone through your 12 cups of water and your appropriate amount of coffee, take a taste of the other 11 cups and have a bit of taste of that, and then taste the first one. And you'll find in the first one you have a lot of oil, and it's fairly bitter, and it's because there is a difference in what is coming off of the coffee beans in that first part versus the entire extraction. So each one of these has a different condition, and it shows that different components come off at different times. And in the next slide, when we speak about how do I go through and talk about extraction with solvent and without solvent, so we're going to go through a pictorial world of what the theory would show. So let's first look at extraction on the next slide and seeing what an extraction is. It separates the desired amount of substance when it is mixed with others. The mixture is brought into contact with a solvent in which substance of interest is soluble, but the other ones are insoluble. What does that mean? It means that when you put in the coffee beans, you're not extracting out all the other material that's insoluble. You are getting a liquid out on the other side. You're using water. In this case, you're getting a solvent of substance of the things that you want soluble. If you were to use ethanol instead of water, you're going to get out a lot more compounds, and they're going to come out a lot faster. The trouble is you're going to be awake, but you're not going to be of much use for the first couple of hours. But you're also taking out compounds that you didn't want. You wanted the water-soluble compounds, not the ones that were in ethanol. And we'll look at why those things are the case. Why would different compounds come out with ethanol versus water? And so now you have these two phases, one phase that doesn't mix and one phase that does. let's look at the next slide to talk about different compounds, different solvents that are typically used in the cannabis industry. You have water, isopropanol, carbon dioxide, you have ethanol, butane, acetone, propane, hexane, heptane. Each one of these has a different criteria on what it will extract. It will have a different reason of what water will extract out versus carbon dioxide or versus ethanol. And on the next slide, when we talk about the fact that you can have multiple solvents, so you can have CO2 with ethanol, you can have water with ethanol, that's called Everclear or some other compound like that. You can have a mixture of different compounds that come out so that you can have a more sequential thing where you can take off water first and then use ethanol. You can have it so that you can take out multiple components based on what you have for the solvent and solute and solid compounds. I can't hear you. There you go.
John.
Yes.
Okay, so just for clarification, because I'm going to act like I am just learning about all of this information, these representative solvents that you're putting on the slide, that in fact is not necessarily what is allowed, just for clarification. So we're not allowing people to use, let's just say, nail polish remover. We understand acetone is generally used for that, but that photo representation, that's not what's allowed. Same with the butane canisters, that's not what's allowed. So I just want to make sure that people understand. And same with the carbon dioxide. Obviously, that's not what they're using. They are just illustrating.
So yes, thank you so much. Yeah, I typically won't use those, but as we see these all the time out in the shelves, and we saw them in the early days of people just using acetone. So each one of these is definitely not one, but you will find them as components out in our everyday life that shouldn't be
Yeah.
used anymore.
Yeah, we absolutely did. And I will say so, as John had mentioned, I've been doing this since 2010. I was the first, but also, did I see these? I absolutely saw these. I saw these containers. So for those of you who are in a market that is still fairly new, you may come across this and you may see things that you think is okay. And I just wanted to make it very clear to attendees and anyone who watches the recording that these representative photos are not approved.
No, thank you.
Continue. Thank you. Continue.
Next slide. And now we talk about separation. And separation is without any other component in between. A typical one of this is the filtering. So there's no liquid involved with the filtering. You're just taking out larger molecules from the smaller molecules. You're being able to separate things that don't have to have chemistry and solvent involved. So if I look at the next slide, it's grinding. Grinding is is mechanical. It allows you to make a smaller and smaller amount of particle size so that you are able to get more material through. You also have chromatography where you're separating by the different types of material. They are going as a solvent with a carrier. So there's a difference between using a solvent as a carrier and using a solvent as part of the separation. And filtration is also, as we talked about, another way of having that. So if you are also pulsing electricity, or you use microwave, or you use acoustic, you're being able to break open a cell wall, but you are not using that as a solvent. So you can have those two involved. If I look at the next slide and I see that I can have different ways of doing that. So I can do acoustic at the same time being able to go through a filter paper. You can have a combination of different things that are already in line. So they can be in combination, or they can be in sequential, or they can be sequentially combined. And then the next slide, when we look at the fact that these two things are happening with multiple solvents and with multiple ways of separation, we also see more and more that these are being combined. We're doing acoustic with a microwave assisted or acoustic assisted separation. So on the next slide, it becomes more and more difficult to say, am I doing an extraction or am I doing a separation? And so I make up two words. It can be either extraporation, because even on the coffee, I have the coffee in grounds in the filter, and now I'm passing the water through, but I'm not passing the other material through. So I have a combination of a filter paper and water, and I'm using them at the same time. They are sequential in many respects because you're taking the water and you're taking the material out, but you're holding back the material you don't want. Or it can be a separation. The same thing can happen, that you were actually doing acoustic and then you're doing at the same time an extraction with a solvent. So it becomes, is it really an extraction or is it really a separation? And I believe in the combination, it could be either one of these words. In the next slide, when we look at that entire process, we can see how this goes back through and correlates all into one slide. So I take this one paper by Eric Lesallier and I go into the next slide to show a representation he has on one of the figures. And if I blow that figure up in the next slide, I now see something that we are now introducing. So when we talk about like dissolving like in the world of chemistry, there's a way of looking at it as called log P. And log P is looking at octanol and water. So you're looking at something that is nonpolar and something that is polar, and being able to look at each one as a ratio to say which one is more likely to come out in this solvent. So when it says like dissolves like, you have compounds that have a specific log P themselves. For example, you can look at the cannabinoids and they're around 5.8. What does 5.8 mean? 5.8 means that I'm more likely to be in the non-polar layer than the polar layer. If I am looking at something less than 0, I'm more likely to be in the polar versus the non-polar layer. So you can start to say, what do I want to have for a solvent that will selectively extract the components that I'm looking for? And then I can see that Twee has a question.
Okay, so again, if I'm just learning this information, from my understanding, from what you're saying, is the log P value will correlate with whether or not it extracts polar molecules versus nonpolar molecules. Is that my understanding?
Yes, it will preferentially go through one. So if I had a great example is oil and water. Water is polar and oil is nonpolar. So if I was to have water and oil in a mixture, and I was to add one of these components, which phase would it end up in? So if it's nonpolar, it's going to end up in the oil phase. If it's polar, it will end up in the water phase. A majority of it will end up in the oil, and a minority would end up in the water.
And something that you and I had discussed when we looked at this slide, I was asking you very specifically about ethanol, because ethanol is used as an extraction solvent in, not only, cannabis and hemp, but many other botanical industries as well. But just looking at this specific slide, I asked you very specifically, I'm like, well, whenever I've seen any sort of ethanol extractions, I see a lot of chlorophyll. I see a lot of chlorophyll in the end product, but when I'm looking at this particular diagram, the chlorophyll is more towards, a higher log P. So how do we explain that?
So every molecule on here, I will say 95% of the molecules on here will show a ratio in the nonpolar and in the polar. And so it may not be all of the chlorophyll that shows up in one phase. It may be a proportion of the chlorophyll. It might be an 80-20. So 80% shows up in the nonpolar, 20 shows up in the polar. And so it's never 100%. Rarely it's 100% that shows up in one phase versus the other. And the reason that is because those molecules will stay around each other. And so that entire phase will be on one versus the other, a majority in it. So even with the ethanol itself, so it takes out all the coumarins and takes out the cannabinoids, but it will also allow you to do something sequentially. So these show a pure ethanol. If I was to have ethanol with water, i.e. vodka or Everclear or something, it's 80% water. So you will have a different extraction based on your power of percentage of ethanol and in water. Remembering that water and ethanol will go together many times. We see that in almost all the bars.
Sure. So I'm seeing also that the anes are more towards the right for the log P. Where would hexane and heptane, where does that fall on the spectrum for log P, if you know? And where would, you know, isopropyl alcohol, would that be close to ethanol as well? And I'm bringing this up because these are, you know, generally solvents that you would see during extraction processes or purification processes, and also where would CO2 fall on the log P spectrum?
Yeah. So when you look at this one here, you'll see the ethanol. So it really is talking about most of the alcohols. So whether it's isopropanol or any of the other ones, you're still going to have it around the ethanol. And the hexanes will be more towards the right side where you're seeing the octane, for example. So octane, you know, there's not the preferred solvent, but if I go back through, I have hexane would be close to the octanes. So they'd be close to the nonpolars.
Very good.
In the next slide, we actually talk about some of the things that you were just mentioning with CO2. And CO2 can change its strength based on pressure and temperature. So as it becomes more and more condensed and becomes supercritical, then you will look at how these change. On the left-hand side, the chart on the left side talks about solubility. And so that tells you how soluble it is in the conditions that CO2 is in. So if I look way over to the left, I see, pressure of 11 megapascals. So it's around 1100. And then you see a continual rise of the solubility of CBD as the pressure increases at this temperature. We'll call it 107 degrees Fahrenheit for being in this world, or 42 degrees C. And so we see that specific temperature that you will have at that specific pressure, CBN will be less soluble than CBD at, say, 15. If you look across the bottom, you see 15, and you look across the top and you see CBD is more soluble, certainly more soluble than THC or CBG. If I look on the right chart and I see that red line, and the reason I have the red line is because it goes to the top of the chart on the left-hand side. So it's unfortunate they're on the wrong scale, on different scales. But now you see at just 10 degrees higher on C, which is 127 degrees, so 20 degrees warmer on the Fahrenheit side, that you see the same pressure at, we'll say 15, you see across the bottom, and you will see that CBN is far more soluble than CBD. So CO2 has the ability of changing its solvent strength based on the conditions that it's in. So when people are going back through and you're wanting to have a separation, you can do a separation based on the temperature, the pressure, and the amount of material you have in.
John, can you explain to those who don't fully understand what a supercritical fluid is?
Supercritical fluid. So typically we're in Earth's atmosphere, we're used to a solid and then a liquid and then a gas. We see those all around us. What happens is if you're able to take a container and hold this material inside that container and you increase the temperature and increase the pressure, you will now move into a fourth phase, and that is supercritical. And YouTube shows these really nicely in the fact that it will go from a gas, you will see it change into a liquid as you increase the pressure and temperature, and then you will see it move into a a supercritical state, which is, it looks like a fog. And what happens though is now with CO2, you will have the oddity that this material, this phase, will act like a gas. So it will permeate into a material, but it will act like a liquid in the way that it takes the material out. So the supercritical with CO2 acts like a gas and a liquid. And then when you see here, you can increase the solubility, you can increase the strength of that solvent based on the temperature and pressure. And you can see a significant difference on the CBN versus the CBD, for example.
Mhm. Thank you for that explanation. And I guess, for those who are in industry, a lot of times, when you have a particular batch, a particular cultivar that you know what the cannabinoid profile is, you're going to make tweaks to how you run that material, what temperature, what pressure, so that you can extract the maximum amount of that specific profile. So if it has higher CBN in it, then you're going to run it differently than something that has the higher THC in it. Correct, John?
Yes. And it's also making sure that your material is the same that you're putting in. Does it have the same um, density? Does it have the same, is it the same particle size? Is it the same amount of moisture? So that you start off with something that's always the same so that you will get the same result. Because same as the coffee, if you have something that has a different grind and you grind it differently the next day, you're going to have a different result. And
Yeah.
so you're right. It's the type of coffee that you put in, and it's the results that you will get out.
Very good. Yes. And what we'll find again in industry for regulators, you may find that they're extracting mechanically separated trim, from when they're trimming the flower. You might
Yeah.
have hand trim, which is like the leaf. You might have other material that maybe it is just pulverized, very similar to an espresso grind, right? So again, you would have to run it very differently. Now, would you also state, even though that butane and propane generally do not act as supercritical fluids, but we will also find that they make tweaks, to the time, the temperature, or even the pressure that they're running it based on the cultivars? Have you seen that as well?
Anytime that you have something that you've changed as a solvent, then you will change your results. Same as the temperature of water. And also having the mixtures. The same as all of these have, you can have mixtures. You can have a mixture of propane and butane. You can have a mixture of CO2 and ethanol. So you can have mixtures that will also give you different results.
Very good. Thanks for that clarification.
Next slide. Well, let's quickly go through what's happening in an extraction. So I have a green thing here, a green circle, and inside of it, I have a cell wall. And what happens is you have compounds A and B inside. And if it's a simple number of 1A, 1B, then the solvent will move through the cell wall. And in the next slide, we see that once it's moved inside the cell wall, it finds the solubility of A and B. So nothing has changed here. We have A and B, whether it's caffeine or theobromine. You're going to have two compounds that have disparate solubility in the solvent they're in. It doesn't mean that they are identical, but now you take this solvent and you carry it back through the cell wall out to the outside. In the next slide, we see that it's being carried off and by the solvent itself, so that you have now collected A and B, and they've moved along the way. So as you have a liquid moving by a particle of some sort, it goes through the cell wall, it finds itself inside the cell, it solubilizes compounds that are inside the cell, and then you move outside the cell and then transport it to collection. That's what you're doing with coffee. Water's going in, it's going inside the cell, it finds the caffeine at a certain solubility, it moves out through the cell wall and comes out through when you're doing the process. In the next slide, you see that it goes away. But if I have a disparate number of A's and B's, in this case, I have, what do I have? I have 1, 2, 3, 4, 5, 6, 7 A's and 7 B's. And if the solubility is different between the B's and the A, that my B is more soluble in the solvent that is going in, ethanol or hexane. So I have a certain solubility of A or B, and I take off 3 A's, 3 B's, and 1 A. Then the next time around, I'm going to take off another 3 B's and 1 A. And when I'm out of B's, then I'm only going to take out A's. So as you're doing an extraction, you're going to have a difference in the concentration based on what it has taken out from the previous time. So if I go to the next slide and I see that I have a different ratio of B's and A's, maybe I take off 5 B's and 2 A's. Well, I'm left with 2 B's and 5 A's. So the next time around, I don't have enough B's to continue. So I will have a difference in my results as I move through the extraction and the number of times that I allow that fresh solvent to go through. If I look at these and say, okay, I have a lot of these samples in the next slide, and I start at the very base of a cylinder or whatever collection it is from the top or the bottom, but I'm going to say that I have all of these compounds and everyone's doing something a little different. If I go to the next slide, no matter what solvent I have that's going in this very simple example, then I have a solvent that's fresh that's going in. And 1 millimeter up, I no longer have a fresh solvent. I have a solvent that has a mixture of A's and B's. And if you take this and think about coffee with 1,500 compounds in it, you have a lot of things that are different, but you're always starting off with the fresh water at the top. So when I went back to my example of the coffee cup and you take out those first ounces, you might have a lot more B's than you have A's. So you have a smaller amount of caffeine, but you have a large amount of oil that has washed off. And that's why when you do that experiment tomorrow morning and you taste that cup of coffee, you are going to be surprised on how bad that tastes, because it's mostly the compounds that you're not wanting. It's mostly the oils. And when you usually had a cup of coffee, then you have that entire mixture together. And so you don't notice it because it's a smaller concentration of the total. If I look at reality in the next slide, reality is these compounds are not in little circles. This looks like a very bad duck. But if you go back through, you have A's and B's in a very irregular cell. And if I go to the next slide and I look at that single cell and I start to follow this very complex set of arrows, if I look at number 1 and follow number 1 through the little circular to the duck, it now goes inside that irregular cell. It sees A and B. In step number 3, it goes back out through the cell wall. And now I have a number of different A's and B's that are already there. And so now it's released and it goes on through to your collection, your coffee collection. Some other water molecules are going to go right by. If the rate of transport is too fast, it hasn't got enough time to get into that complex. So you're going to have a weaker solution. When you look at your coffee maker and it says strong or weak, the only thing it's really doing is slowing down the flow rate of your hot water. If you look at it, it's slowing down the flow rate of your hot water so that it can get into more cells and it can get more compounds out of those cells and then being released into the stream. The way to look at this another way is also when you talk about why are some extractions in the beginning. So when you are grinding, for example, a cannabis plant, you are grinding it, you are breaking up some of the trichomes. You're still going to have some trichomes in place that you're going to do this type of process where it goes into the cell and then collects and then goes outside of the cell. But many times you've already released most of those compounds. You've broken up the cell wall. So those compounds have all fallen out. They're out in the open. So if you have a situation where you have a moving walkway, which will be our mass transport, and you have an opportunity to go back through, and you as an individual can go into a house that you're supposed to collect dogs in, then you get off the moving walkway, you go into the house, you find the dogs, you're able to carry one Cocker Spaniel, you're able to carry two Shih Tzus, and a Pekingese, and that's all you can carry. And there's only so many Pekingese that are going to be in the house, but now you go back out to the moving walkway with these dogs in your hands, and you move down and you drop them off at the end, and then you go back around. If it's a recycling world, you go back around again, you don't have any dogs, so now you're going back and you find more dogs, and each time you're able to find the number of dogs that are in each house, and you go to each house and then to the next house. Those are happening with a lot of people and dropping it off. If, however, in this analogy, and I use it crudely, but there are no animals that are hurt in this analogy, and you blow open the house so that the dogs are all out in the front yard, well, you're not having to go into the house. You're just collecting the same number of dogs. You cannot collect anymore. You're only a Cocker Spaniel, two Shih Tzus, and a Pekingese. You are only able to collect those, but you're able to collect them as you're walking through the process and down the moving walkway. But is that faster than going into a house? Number three, I may be on number three. What if it is a very large apartment building? When you go into the apartment building, you go to the first floor or you go to the ones that are closest to the door, you grab those dogs and you go back out until you have collected all the dogs on the outside layer before you go inside, even further into the cell or even into that matrix. And so now you have capability where you are getting slower as time goes on because it takes more time to get deeper into the apartment building. These are the things that happen in extraction. These are the things that why there is a difference between the rate of extraction that you see and the rate of collection that you get. And that is why they do acoustic. That is why they do electrochemistry. That is why they do microwave. You're trying to break open the cell walls so that you can more quickly access the materials that is soluble in the solvent you're using. What has this got to do when you take water versus ethanol? Ethanol you can grab a lot more molecules. It has more solubility of the molecules that we are interested in the cannabis industry currently. That doesn't mean that you're not collecting some alkaloids or some other compounds. It's just that in this solvent, you're collecting most of those. If I go to the next slide and I look at how this actually makes an imprint on what we're doing, it means that how we're collecting and the rate of collection. On the next slide, when we look at why this matters. So now I go to the processes again. Why? What exactly is being controlled and how do you know? Why are these limits selected instead of others? Why am I using a certain percentage of water versus ethanol? Why am I using hexane? Why am I using CO2? Why am I using butane or propane? Why am I using those solvents to be able to have that? But also, what is the limit on what I have for the size? When I look at the evidence of what remains in control, are they in control? What happens when something is not in control? That allows you to understand what's understood and bonded versus just merely repeated. And it allows you to say, okay, they don't really understand this process. Tweet looks like she has a question.
No question, just a comment. And, for regulators, what we learn is you can trust what someone says, but you have to verify. So everything that you're saying now is just, hitting home with just the trust but verify. You have to ask them, right? And you're absolutely right. A lot of times the operators don't fully understand what they're doing. They're just doing what they're told to do.
And so something goes out of control with that operator without no one there. Now you have something that is an out of spec and may not know that it's an out of spec and why.
Mhm. Yeah, good. Thank you.
In the next slide, I look at the facilities. And you have the same sort of questions that we'll look at as we move through the trilogy a little bit closer and as we move to the next three. And that is, all right, now I have equipment. What's the appropriate equipment for the material solvent? So you have different components that have to be able to be used for what they're used for. And so you need to have the right seals in place. Is it the right seal to be able to use with this solvent? Can it be cleaned? How can I know when it's clean? How do I know effectively that it has been cleaned? How does the layout prevent contamination and mix up? How do I know that I've gone from a grinding room over to another room? You don't want the grinding to be in the same room that you have the extraction, and you want to be able to have the right gear to be on that you're doing grinding, so you're not getting material into your body from the fine grinds, and what risks are out. What risk has been designed out of the process, and one way to do that is to have grinding in a separate room and then bringing the transport in a compliant way. So now you see the facility design, you see the component design, and so when you have a heavy reliance on SOPs, standard operating procedures, it doesn't compensate for poor physical design. Tui has a question.
Yeah. So I just wanted to make a comment regarding facilities and equipment, specifically with very large extraction equipment where it's generally going to be clean in place. Generally, you'd have to fully understand what the manufacturer is making recommendations on for how to clean this equipment, but also how do you verify that it's clean, as Dr. McKay had already mentioned. Now, I will tell you a story of a time when we were using it was a CO2 extractor. It was not a water CO2 extractor, but it was a CO2 extractor. And it was visibly dirty. You can see, the accumulation of oil from the CO2 extraction. But then we used an ATP swab just to verify whether or not it was actually clean, and it was the vessel that was, holding the plant material, and it had not been cleaned post extraction and had been sitting there for quite some time. And so, of course, I would assume that it would, be off the charts for ATP to verify that this is not clean. But lo and behold, it did not register. It was insane. I didn't understand it. And so we thought maybe the swab was wrong. So we tried several swabs and we swabbed the same area, again, visibly dirty, but it registered clean. And the only thing that I can possibly think, the reason why it registered clean, and John, you can tell me what your thoughts are, was that possibly the level of pressure that was used in the CO2 extractor, like almost obliterated, I'm guessing, the ATP, so that it didn't register, because that's what the ATP swabs do. They just kind of measure how much ATP is there, live or dead, right? So that was something that I had never fully comprehended, but I wanted to bring that up specifically about, you know, uh, facilities and equipment and what their SOPs are, and how do you verify when you can't generally rely on the same types of techniques and technologies that you will in regular food manufacturing or botanical manufacturing? Like, what would a regulator do to verify that it is in fact clean?
What's ATP?
Is in all living cells. It's in
Okay.
all living cells and how they determine whether or not, let's just say, for instance, if I am trying to see if there is any residue from a manufacturing operation in a piece of equipment that is clean in place that you can't fully disassemble, and maybe there's a biofilm of whatever bacteria inside, and we're just going to swab the area to see if there's any sort of ATP being registered. And
So
that's,
yeah,
yeah.
it wouldn't live through the pressure and temperature. That cell wouldn't live. So inside that vessel, it's not there. But inside another vessel, so if, for example, you have aflatoxins or ochratoxins, so when you're going back through and you look at um, what can be done with a mold will make a molecule. And so therefore what it does is you might have compounds that are made before you killed it. And so you're still going to have ochratoxin, aflatoxins on the outside, but not on the inside. Once you've got it into that CO2, for example, when you're using that example, there's no way that those guys are living through that pressure and temperature. It's just, but that's on that side. It's on the extractor side, but on the collector side is probably where you would have found it.
And that's, when I'm speaking, this is again very specific to CO2 extraction, not necessarily another solvent. And then also for clarification for the residue, I mean oil. Like you could see the sticky oil on the
Yeah,
vessel on
yeah,
the inside. I'm touching it with my hand. It's sticky and it's brown, but then when I swab it, there's nothing, absolutely zero. And it registered as clean. So
yes. No, no.
interesting.
So when I look at the next slide, I talk about materials and process. If I look at the materials, again, it's, how do you get trace material? How do you find it in the final product? Where could material be lost? Where could it be contaminated, as Twee was talking about? And how do you control in-process material and waste? What happens when you have the waste, and what happens to the material that fails inspection? And we will look at that process a little bit on what can happen. And so they're finding out, where does it go and what do you do in between? There's spent material, because it's still going to have some material in there, and it's also going to have, which is different from a raffinate. When you do a wash in your washing machine, and more recently my washing machine motor decided to stop, as far as, it still would go back and forth and fill, but when it empties it didn't do as well. And so when I look at, and when I was having to pail out my washing machine, I could see that there was a lot of dirt in there. But in that case, I'm not keeping the extract. I'm throwing the extract away. And so the clothes are my raffinate. I'm cleaning the clothes. But on the other side, I have to find out what happens. Just one moment. I have to switch a slide.
So while Dr. McKay is switching his slide, if you have questions, please make sure to put it in the Q&A chat box, and we will try to answer it either live, or we will answer it at the very end. I know this is a lot of very dense information, but hopefully we are making it bite-sized and digestible for everyone. So please remember to add all of your questions to the Q&A.
Good. Thank you, thank you, thank you. So if I go to the next slide, and I talk about documentation and records, you have to have good records. Where are they? These are good questions to ask, and we'll ask these during the trilogy, and not so much today, but you have to be able to have records and know what was done and what wasn't done and how it changed. And when I look at the last part, where I take the whole slide and I look at everything that's involved on the next slide, safety and risk management becomes everywhere. Where are the hazards that exist in this process? Are they controlled? Is it paperwork? What happens when systems fail? What happens when I go out of spec? What do I have? And being able to know what I should be doing next. And we'll cover some of these in the trilogy in the next couple of seminars. But on this one, just being able to look at this and being able to know what those risks are, and we'll look at those in another presentation. In the next slide, when I look back at what we're actually concerned with most of the time, is we're talking about the cannabinoids, but this is only one piece of it. And the cannabinoids exist in different secretory cells, and they are made through the process of the biosynthesis. And over time, we've gone through to know more and more about these, as well as how they are biosynthesized. And in the next slide, we see that there's different types of trichomes. And so even within that, with that criteria, being able to know where most of the trichomes are and why you don't get 100% extraction is because there's different plants, there's different parts of the plant, and being able to extract those in a timely fashion. If you went forever, perhaps you could get them, but there's no reason to be going through that part as a manufacturer, because you're now costing more money to be able to extract the last part. When I look at the next slide and I decide, okay, I've gone through a field, some miracle happens, and now I have an oil. I hope that through some of this work that we talked about today, that it's not magic. It's just a scientific process, and a scientific process that works through many natural products that we're doing extraction from, including coffee, including decaffeination of coffee, including some of the other decaffeination of teas. So we're trying to be able to have different processes in place that allow us to have the concentration of what we want. And when you look at the next slide, to say, all right, how does that actually happen? Remember where we came from. We came from trying to be able to assure that we had the concentration. We wanted to make sure it was efficacy. We wanted to make sure that there was safety and testing. So each one of these parts becomes critically important for the evaluation of compliance. Why is this happening? What is an effective concentration? When I look at the next slide and I say, all right, so that makes sense. Why is it so fast in the beginning? Why does it taper off to the end? We go back to the dogs. The first part of the dogs is in the constant extraction rate. It's mechanically dominant. It means that everything has been, there's been some rupture. And so the material is outside the cell wall. When you look at the last part, not the one in the middle, but the last part, that is when it's thermodynamically involved. It is going into the cell, finding the A and B, and then coming back out of the cell. It takes time, but it also takes energy. So the first part is very fast because it's free. It's really not inside the cell. Most of it is not inside the cell. A lot of it is outside the cell. So now you're just doing solubility. The part at the end, it's inside the cell. They are both happening at the same time. And so when you have the rate in between, that's when you start to see a tangent of you're moving to the next phase being the majority. When I have the next slide and I look at what does that mean as I'm determining through the process of analytical testing, then I can look at that test and I can see that the different variables will change as the extraction is going on, much as we saw with the CO2, where I could have a dominant first part of CBN versus CBD. But I can also see that as I'm moving through the process, I will have a different concentration based on remembering how we brought out most of the B's versus the A's because of solubility. Once all the B's are gone, you're going to find that now you're starting to take out the A. So you're going to have a difference throughout the concentration of the material based on where it is in the extraction. So you're going to see a change in the ratio of these components. In the next slide, when I see, okay, how does that look from a point of view of does that really happen or is it just a mystique? From the left to the right, is a vessel that was with CO2, for example, and you can see on the left-hand side it's very light. On the right-hand side, you can see it's dark and has some clumps. It shows that you're being able to have material that started from the bottom in this case, and it wasn't fully extracted. It was extracted about 2/3 of the way, but you can see the top part is not. When I look at this tube in the next slide, where I have a line down the middle, I'm now doing a cross section, and you can see even through the cross section of this area that wasn't fully extracted, it was on its way but wasn't completely extracted. You can see that there's a difference in the shading. The middle is much lighter than the outside on the top side. And so you can see even within a cross section that you don't have a full extraction. Now, when you go through a compliance to say, okay, what do you do with your material? How do you know that you've got the result you wanted? These are the types of things that you visually can see that it wasn't fully extracted. If I look at the next slide and I think, okay, if I was to look at each one of these components in 9 quadrants, how much do I have in each quadrant? The picture on the left-hand side is what we had from before, from the bottom of the vessel to the top of the vessel. And the liquid was going, the CO2 was going up to the top. You see that on the bottom, the light material shows you with the cannabinoids, it was extracted. There's nothing there. And then you can see in the middle that on the right-hand side, it has the 1.8% was left. And on the other side, it had the 10.5%. And we saw that when we saw the cross section. And then you look at the top and you can see that you had material still in there, and it shows on the dark green. You have material that is left over. All right, so you have material that's left over, but you want to be able to take this material and be able to extract it all. But how do you know? And those are the compliance parts of asking the questions. How do you know what was in the material? And if you're throwing this material away, it still has a lot of THC in it, for example, in this case. But how do you get rid of it so it's safely not being used, and making sure that all the other material is in place? How do you get to the next part where you say, when you go back through, how do I know that I had everything out? If I go to the next slide, and then I'll review, what happens when I have a material that now I have changed the method, and I go and I try and say, all right, what's the impact of this on the value? There's a lot of value that I'm throwing away. I will go to the next slide. And now what I've been able to do is I said, okay, I see that I have this problem. How do I remediate it? And this is a case where we have now remediated what we didn't have from before. And so now you have everything is extracted. To be able to know how to do this is important to say, okay, I have a problem in my method. Something has happened, and you're able to correct it. So from this part here, it's a correction amount. If I go to the next slide and I go back to where this mattered and what we will cover in later in another presentation, these are the key factors. If I go to the first one, the next slide, I see, okay, these are the questions, but over on the right-hand side, I've shown the slide that would allow us to review, why is this an impact? If I go to the next slide and I look at the facilities and equipment that we will cover in another presentation, I can see, okay, that shows me what happens. What happens if I have a scale? What happens if I'm changed my conditions? What has happened to my extraction? What has happened to my results? In the next slide, when we cover things in another presentation, we talk about materials. How do I know when I'm done? How do I know what's lost? How do I know what's contaminated? How do I know how to get rid of it? That shows what we'll cover in another presentation in more detail. And the next slide talks about what I have for known. How do I know that I've reacted to that? Because I've done testing. I've seen what's the consistency, what has happened in each one. And the last one going through on the next slide shows what I have for the safety. How do I know where everyone is? And how do I know what I have to be able to do to correct it. And finally, that's everything that's a safety and risk. On my next slide, I put some of the information that I had for myself to be able to go through this first part of the trilogy of the overall basics of extraction and concentration. One thing that I would say is, as we look at this, what are the principles? And to go back through and always think about coffee. What have I done for a grind? What have I done for espresso? What have I done for the amount of caffeine in each one? Each one of those is important, and being able to now correlate it to what you have for cannabis, but also on the compliance issue of manufacturing. Tui has a question.
No question. I'm just letting you finish out so that we can ask any questions if we need to. We did have one participant that asked very specifically about rerunning the material when you see that, it doesn't fully seem like the solvent has fully transmitted through the entire vessel.
That is correct. And what they did is they didn't, let's back up. When they packed the vessel, they did not pack it correctly. And the second one was they were using the wrong type of method. It was going too fast. And so it went past the other material that was up on top. So it had saturated itself. They also, based on that, then there wasn't enough time. But how would you know there was enough time unless you realized that you had unpacked it incorrectly?
Yeah. And, a lot of times it depends on the manufacturer. Some manufacturers will, either number one, allow for the solvent to soak before they actually extract. And again, it could very well be dependent upon the type of solvent as well. I know that with butane and propane, generally they allow for it to soak for a certain amount of time before they um, uh, start to recover the extract. I'm not sure if that's something that can be done with the CO2 technology. That's something that you can address, John, but also sometimes they will rerun it, or they will even extract with a secondary solvent following that as well. So it just depends on the manufacturer. But for regarding that specific material, could they allow for that CO2 to soak under that immense pressure? Would that vessel hold it for that amount of time? I'm guessing yes.
I'm guessing it can hold it for as long a time as it possibly can, because most of the CO2 vessels are up to 35,000 psi. You're only using 5 or 6, and so 1,000 psi. So on that side itself, that was how they did it, and so they were able to soak and then evacuate, and then soak and then evacuate, and so the best way to be able to do that is to put that in process and put it in line so that it's constantly doing that.
Of course, we're speaking in terms of, having the not only the highest yield, but probably the best yield. But when you're talking about manufacturing, you're talking about time, you're talking about, the labor costs, you're talking about a lot of different aspects. So they may decide it's not worth the time and it's not worth the labor when they run the cogs, right, for this specific extraction. So it's either you tweak what you're doing so that you can have the maximum yield in the shortest amount of time for the dollar amount, or you can do it from the scientific perspective and say, well, this is ideal, right? Because we'll see that a lot in the industry as well. So we don't see any questions that haven't been answered already in the Q&A chat box, and we are at 4:30 right now. I'm looking to see. So Amber's saying benefits of double and triple running. How many runs per column or just risk in one? Again, it depends on the manufacturer. Just run one or multiple. So again, depends on the manufacturer because it does cost time. It costs money. The solvent costs money. The labor costs money. The machine, you've already paid for the machine, or you're continuing to pay for the machine, right? But with the CO2, are they recovering that CO2? That CO2 that they're using, or are they emitting it into the environment post extraction, right? So it really depends. And we're not going to get into it, but that's off gassing CO2 gas, right? greenhouse gases. So, so there's a lot of different methods that people will decide to use, and it's very important to, address it not only as, let's just say, from my perspective. I worked for the health department, so I did inspections, but by doing so, I worked very closely with the fire department, and I worked with waste management. You have to start working with the various agencies that address different aspects of the operation as a whole, to fully understand what the repercussions or ramifications are, for the technology, but also for the process itself. Wouldn't you agree, John?
Yeah, that's exactly the case. So a lot of it can be automated and sometimes it's not, but we'll go through some of those nuances in the next presentation.
Yes, in the next presentation. So you did a great job getting through all those slides. I hope, this for the participants was easy to understand. Dr. McKay is, very great with all of his examples to make it easier for, the lay people to understand the science behind it. And, of course, we would love for you to join us for our next session. We will have that. And sorry, Amy, can you go back to the previous slide? The previous one? There we go. Oh, guess it's not showing. There we go. So, part two of the series is going to be on March 13th with Dr. McKay, and he's going to continue addressing and teaching us about the principles and practice in assessing compliance in cannabis manufacturing. We are trying to keep it in a two-part series. However, if we find that we need to add on a third one, we will do that as well. And please come with all of your questions. If you have specific questions about an operation, you can feel free to email it to us, and we can, talk about specific examples in the next session. And like I said, if we have to extend it to a part three, because that was what the initial intention was, then we will go ahead and do that. But right now, I have John committed for at least two parts with us. So go ahead, John.
Thank you. That's it.
Yes, thank you. And Amy, next slide. Jonathan Gerhardt and myself, we are the co-chairs for the Cannabis, Hemp, and Natural Psychedelics Committee. We do hope that you join us for our committee. It is free to join. Of course, we would love for you to be able to support AFTO. Next slide, please. Support AFTO by not only joining, but joining AFTO as well. We are trying to put on some great, amazing educational material that can be shared not only live, but also recorded on our AFTO YouTube page. So please subscribe to the AFTO YouTube page, where you can find, this recording as well as the previous recording, and all future recordings. And if you have any questions or comments or even recommendations on different perspectives that you would like for us to address or find speakers for, then we are happy to do that. And I'm going to make sure that we don't have any other questions before we end the session. Any last thoughts, John?
I think one of the things to do is to have people understand that it's can be understood, and use principles that we constantly are using to be able to do concentration every day. I think that there's a great opportunity to further educate and make people understand that chemistry is fun.
Yes, chemistry is very fun, and thank you so much for, pointing out the things that we really needed to point out. And one thing that I did want to point out before we end the session was nomenclature. That was, one of the early slides that you had. I address this every single time, every single state, and every organization that I've sat in on meetings. Everybody has different nomenclature, different definitions, or not clear definitions. And so in order for us to move forward and to standardize, we have to have a unified nomenclature and definition, so that we can not only understand how we are regulating, but are we speaking the same language? Do we understand, what it is that you're trying to explain? Just like industry has a lot of different terminology for various types of processes and various types of concentrates, but they don't always align. They may be saying the exact same thing, but they may use two different names to name the exact same thing. So, one thing that, I would like to emphasize again is, we really do have to have a unified and standardized nomenclature and definitions for us to move forward.
Thank you.
So thank you. Yes. And anything else, John?
No,
Very good. Well, we welcome you next time for your part two of the possible trilogy series. And please come back and join us again on March 13th. Thank you, everyone.
thank you.